in their search for the virus, they were aborting fetuses and butchering them. He put a list of medications at the end of that blog post on that got a lot of attention. And he put things on there like aspirin and Tums and Preparation H. The most preposterous of the bunch, though, was Tums, neonatal infant formula.
Mm-hmm.
What they did to develop that was horrifying, because we have cosmetics out there that say no animals were harmed by this product. Okay, why can't we have no human beings were harmed?
Hello and welcome to Living a Culture of Life podcast by Human Life International. I'm your host, Colleen, and I'm joined today by Jose Trasancos. Welcome. And you are the director of Children of God for Life. Is that what your official position is?
That's correct. I guess the title on the paperwork is CEO and chairman of the board.
Okay.
We're a small shop.
Can you then just explain to your listeners how, because I know that Debbie founded that organization. So can you explain how you got involved and what your organization actually does?
Yeah, it's an interesting story. Our connection with Debbie Vintage goes back to about 2006, 2007. My wife actually reached out to her because we were in the middle of growing our family. We have 7 children, and the issue of vaccinations came up. And my wife is one to research everything. She's very thorough, and she's very concerned about anything that has to do with the safety and well-being of our children. And she came across Debbie Vintage's name in an online search. So she reached out, made contact, um, uh, got the information that she was looking for. We discussed it, we made our decisions, and we moved on. But she kept in touch with Debbie over the years. And 3 years ago, Debbie was thinking about retiring. And Debbie wanted to make sure that the organization went to folks of like mind, similar inclination, and would take it seriously. So she contacted my wife, and originally what she wanted to do was to move it to the Diocese of Tyler. My wife was working for the Diocese of Tyler at that point in time, and
Tyler, Texas.
Tyler, Texas. Yeah, we're located in Tyler, Texas, or in the Tyler area. But we discussed that a little bit, and having it be a formal part of the diocese it seemed like it had more problems than problems solved. It would become then a part of a larger organization that would make it more difficult for them to plan. What Children of God for Life does is very different than what a church organization would do. So we decided that we would take it on individually. So we did the paperwork. We redomesticated Children of God for Life from Florida, where Debbie lives, to Texas, where we live. And the rest is history. At that point in time, I was semi-retired, kind of sort of retired. Now, not in the least.
Yeah, it's hard to retire when you work for organizations like that. So, can you explain for our listeners what Children of God for Life actually does?
Children of God for Life is we changed the mission a little bit, because Children of God for Life, prior to us becoming formally involved, was focused on principally vaccines. And we wanted to broaden the scope a little bit and deal with elements of aborted fetal tissue in, broadly speaking, science and commerce. So that's our approach is a fairly wide one at this point in time. We look at vaccines, certainly, and we can talk about why vaccines become such an important issue in a moment. But we're also looking at other aspects of fetal tissue research. A lot of it is understanding human development. There's always this promise that further research along these lines is going to cure just about everything that happens, all the way from pediatric diseases to end-of-life issues. Um, so the use of aborted fetal tissue is actually much, much broader than
Mm-hmm.
vaccines. Um,
Well, Well, like I was saying
So
at the beginning of this, we even have the ebook on the cosmetic side of it. So yeah, I've
yes,
delved a little bit into it and done a little bit of research about the different aspects that goes into, which we'll definitely get into in the podcast.
so we do a lot of research individually and collectively. My wife and I work together on this, and whatever time we have left over, we focus on advocacy. So we sponsor a teen leadership camp that's an annual event, and we're trying to attract um, uh, younger people into kind of the pro life issue. And the theme this year was to know your human dignity. Because a lot of the issues that we deal with are either directly or indirectly related to human dignity.
Yeah,
So we do the team leadership camp. There's legislative outreach. Uh, we had a bill introduced this year in the Texas State Senate that unfortunately never made it to the floor. There were all sorts of issues in front of it, but we're going to try again in the next session. And if we could possibly get a similar bill like that in another state legislature, we certainly will.
that's really
So,
important.
oh, absolutely. Knowledge without application really isn't much use. We do a lot of the research bit. And we focus on high quality research. My wife and I are both academics, or she's an academic. I have a PhD. There's a difference. But we both know our way around scientific literature and academic literature, and that's where we take everything that we present as authoritative has peer reviewed references associated with it. Anything else that might be considered speculation, we brand it as such. And if it's opinion, we brand it as such. But our research is done in compliance with academic standards and therefore reliable. Now, it doesn't mean it's right 100% of the time, because sometimes, as we learned during COVID, the scientific literature is not always reliable.
Yeah. So do you have a PhD in like the science field then, or like do you have a scientific
I'm
background
a
for this?
mathematician. I'm a
Okay.
mathematician. My academic background is in applied mathematics. I also have advanced degrees in applied analytical methods and political science.
Okay. So you understand how the numbers. So do you have any kind of scientific background for going into this then, or are you just really good at being able to logically look at the like research and figure it out that way?
Well, the logic piece is very important, but in my postgraduate studies, I worked with my share of physicists. Uh, Much of what's happening in the medical field is biochemical, and thankfully, my wife happens to be a PhD in chemistry. So if I run across something that I really need some help on, help is only hey honey away.
That's awesome. So, what is the history then of the fetal cell tissue research? Can you give a little background for like listeners? So, I know some people might have heard about it in vaccines before, but just I know it's a topic that is getting to be more common, but most people don't really understand that much about it. So, can you just give a little background for our listeners?
For all practical purposes, vaccines and fetal tissue have been have been kind of joined at the hip from the very beginning, and this dates back to the 1930s in the development of the polio vaccine. Uh, the polio vaccine um, development happened in the mid to late 1930s. The Sabin vaccine was one of the first broad application vaccines that were developed. And polio was a particularly concerning disease. It was communicable. It also had impacts that were moderate to severe, and the severity of it was actually significant. So there was a great deal of interest on developing something that could be a prophylactic to poliomyelitis. And the first step in that was the isolation of the polio virus. And um, babies were butchered. I'm sorry if I pause on occasion because this is kind of difficult to talk about. But in their search for the virus, they were aborting fetuses and butchering them, looking to isolate the virus. The reason they were using fetuses was the infection would pass from the mother to the child in the womb, and in the womb, that infection would be relatively unmolested from other confounding elements. And some of this had to do with women's hospitals that catered to the indigent and to the quote unquote feeble minded. There are documents out there and presentations that we have on our website that give the details on this. Excuse me. But also descriptions on how the aborted fetuses were obtained.
I'm
And
guessing this was
most
mostly illegal abortions because obviously abortion wasn't legal at that point. So they were illegally basically aborting children and then using their bodies.
these were correct. Yeah, these were in the terms of the day medically indicated for a variety of reasons. So the connection between these abortions and feeble-minded patients, and I find it distasteful to even use that
Mhm.
language, but that's what's in the literature. Um, was there was an official capacity associated with the ordering of those abortions, and most of the abortions were hysterotomies, a cesarean section. And the language describing the obtaining of these specimens was really kind of disturbing, where they would I'll paraphrase, but they would say that the fetus was obtained through surgical hysterotomy, and whenever possible, the fetuses would be kept inside the amniotic sac, placed on a sterile towel, and dissected as quickly as possible. So what they did was they delivered a baby by cesarean section, and the baby died during the dissection because they were, yeah.
That's
Um,
yeah.
and that's why I say babies were butchered during this process,
Mhm.
and that was how they isolated the virus and
Did they have
the
did
development of
they
the
quote
bacteria.
unquote have to? Like were they choosing to do this on unborn children because they wouldn't be affected by any other viruses? Was that the logical reasoning for using unborn children?
Yes, and they didn't have the benefit of what we know today. Were there alternatives? Knowing then what we know now, absolutely there would have been alternatives. But medical science, um, ethically and unethically, has developed quite a bit in the last 90 years.
Mhm.
And the fact remains is that babies were butchered in the process of developing the poliomyelitis vaccine,
Wow.
the Sabin vaccine.
I never knew that. I've read more about this stuff later on. People talk about how I forget which cell line it is that started in like the 60s, 70s. People talk a lot more about that. I didn't realize it went all the way back to polio. That's pretty harsh.
Yes. Went back to the 30s, and that was kind of the origin of it. And the development of the Rubella vaccine was generally similar, but the Rubella vaccine came 2 decades later.
Mhm.
Um, and there were a number of infants, fetuses, babies that were aborted in the same manner for the same purposes in looking to isolate the rubella virus. And that virus passed and what made that virus particularly important, at least in the eyes of the researchers, to get their hands around, was its ability uh, to result in severe birth defects to born children. So this is kind of the emergence of a remarkable irony,
Mhm.
um, where we exploit and kill the unwanted for the benefit of the wanted. And the use of aborted fetal tissue in biomedical endeavors really just does come down to that.
What is it used for in today's day and age? Are they, because I know that it's used for more than just vaccines now. So how is this the tissue of children used in modern science?
Well, I guess before we go there, I think we should talk a little bit about what cell strains and cell lines are
Okay.
and why they're valuable. And valuable from a scientific standpoint, not valuable from a moral and ethical standpoint. But a cell strain is is a group of cells that continues to divide naturally. Now, a lot of the biologics that are used today in biomedical research are in fact cell strains. And what distinguishes a cell strain, primary culture, from a cell line is that a cell strain has a lifespan. And one of the first cell strains developed was Wistar 38. And the Wistar Institute is located in Philadelphia, and it was developed by a doctor by the name of Leonard Hayflick. Leonard Hayflick did a great deal of research in this area, whether it's cell lines or cell strains. He also he also authored what is known today as the Hayflick limit. He looked at he examined how cell strains, primary cultures, divided over the years, and when that division rate started to decline. And it actually makes perfect sense when you say it this way, that the Hayflick limit says that a cell strain will live about as long as the average lifespan of the donor organism. So if you have a cell strain that's human in origin, you figure those cells are going to replicate in a fairly healthy and normal fashion for about 70 years. And then it starts to fall off. Ultimately, the cell strains, the telomeres, every single cell division, the telomeres get a little shorter and a little shorter, and that's our genetic time clock. Um, so cell strains aren't indefinite. There's always a need for more, and Wistar 38 was established back in in the early 1960s. So it's reaching the end of its life. So some research is going into how they're going to replace it. And that's what a cell strain is. It's a primary culture. It's human cells that are placed in a nutrient-rich solution. They continue to divide on their own, and at some point, they expire just like we do. A cell line continues to replicate indefinitely, and that's a cell line can be taken from like HeLa cells. The HeLa cell line has been replicating for decades and decades and decades with no signs of slowing down or anything like that, because the source of those cells was a cancerous tumor from a woman named Henrietta Lacks. HeLa, He stands for Henrietta, La stands for Lacks. That was the origin of those cells. And the cells that were taken from a biopsy from her tumor were, without her consent, cultured and established the HeLa cell line. Shortly after the biopsy, she succumbed to the disease, but her family has come back and said, you've appropriated something from Henrietta Lacks. We are her next of kin, her surviving family. You owe the family some compensation. So it's kind of interesting that those cells from a biopsy established a cell line that turned out to have a significant amount of commercial value. And it's recognized that without recognizing that value, that there is exploitation.
If it was from a cancerous tumor, wouldn't the cells be cancerous then?
They certainly have a predisposition to mutation,
Okay.
and that's what happens with cancer. But that's that's what makes cancer so dangerous. It replicates at a remarkable rate all on its own, and it's very, very difficult to stop. So, um, cell lines that have been established from cancerous tumors or pre-tumor masses, um, they, um, they require no further work other than to say there they go. Now, the most commonly used cell line that's fetal in origin is called HEK 293. Human embryonic kidney is what HEK stands for. 293 is the 293rd iteration of the experiment that ultimately established the cell line.
So the 293rd child, basically.
It's unclear how many children were involved, in taking the samples,
Okay.
but absolutely, almost certain, there were multiples. So it's not correct to say that there were 293 children. It's also not correct to say that there was one.
Okay.
But you have people on both sides that are just kind of arguing. The truth lies somewhere in the middle. Um, and the number of children that were involved in establishing HEK 293 um, probably wasn't close to 293, but it was certainly into the dozens because a child can produce organ samples more than one organ sample per child.
Okay. So it just so happened to be a kidney. It wasn't that they were taking kidneys from multiple, like they looking for it from any organ and it happened to be a kidney, I guess.
It just happened to be kidney, and the organ source is important because organ cells are very dynamic little factories. They're very, very busy from a biological standpoint. They actively metabolize their food at a high rate. They produce their own energy. They are enormous protein factories. The human cell is really a remarkable piece of divine engineering. I mean, um, uh, everything that goes on inside the human cell is just spectacular. Um, and cells in organ tissues like kidneys, the liver, heart, less so heart muscle, but certainly the kidneys, liver, and lungs are high on everybody's priority list because the cells in those tissues are very, very busy. They have high enzymatic content. They produce a wide variety of proteins. They can be modified to enhance certain expressions, as we can talk about in the context of HEK 293. Because what made HEK 293 immortal, and they call it immortal, which is another just remarkable irony. By modifying the genetics of that cell line with DNA from an adenovirus. They genetically modified the cells, the kidney cells, from cells from an adenovirus, the adenovirus 5, I believe, and that modification resulted in perpetual, or at least it's thought to be perpetual replication.
Okay. Interesting. So that's why it's called an immortal virus because it doesn't have the time limit that a cell strain would have. It will just keep producing.
Correct. It's thought that these cell lines will replicate indefinitely.
So then how are they used?
You and I will never know, but
Yeah. How are they used in modern science now? Because I know it's like I said, I know that it's broadened out beyond vaccines. So how are they used in today's day and age?
Well, it goes beyond vaccines and into into a variety of other medications. And what makes them useful is the cells have expression characteristics. There are bodies within the cell whose job it is to produce proteins, and those proteins are a response to something. Um, and among those proteins are things like antibodies. Um, and our immune system produces antibodies in response to a great many things. So the connection with vaccines is clear, but a lot of the other proteins that a cell can express don't have to do with the immune system. They have to do with the body's own physiochemistry. So if we're talking about a disease or a condition that is the result of some dysregulation in the body because you have too much of this and not enough of that, um, that can be controlled through expression of proteins at the cellular level. So the utility of fetal cell lines is actually much broader than vaccines and anything that's related to the immune system itself. Anything that's regulated by the production of proteins at the cellular level has a potential application with cell lines,
Interesting.
and that's a lot.
So it probably does that mean that this now affects a lot of the medications that are available out there in the market today? And is there a good way to figure out which ones those are? Big
Um,
question.
a good way, a good way, no. A way, yes. It really is pick and shovel work, um, and I'm not sure how one could do that without the internet,
Mhm.
because the answers lie in the scientific literature, and sometimes it's not straightforward. Where you can read the materials and methods section in a peer reviewed paper and not find anything remarkable, but you can find a reference to another paper that has a reference to another paper that has a reference to perhaps yet another paper, where you find the specific method that was applied in the original paper, and that specific method made use of aborted fetal cell lines. Um, yeah, it is knowable, but not easily knowable. It is not easily discoverable. If you don't know how to navigate the scientific literature, um, and if you're at all intimidated with certain types of terminology, it's very easy to throw up your hands and say, "I'm done. I can't figure this out." But the information is out there.
How do people navigate that when it comes to picking medicines and stuff like that? I know that you've done some research and you have lists of the vaccines on your website that people like are ethical versus ones that aren't ethical. What do you suggest
Yeah.
for people who want to navigate this and want to avoid medicines that are connected to these cell lines?
Well, yeah, there are some rules of thumb that I let some people know, because some of the vaccines that are in common use today aren't really concerned with viral pathogens. A virus um, cannot replicate on its own. A virus needs a host cell. Uh, and if you're talking about a viral vaccine or a viral medication, something having to do with a viral infection, the chances of that involving fetal cell lines are are very much higher than something like diphtheria, tetanus, or pertussis. Like the those are bacterial pathogens.
Mhm.
And bacteria don't have a problem at all replicating on their own. All they need is food.
Mhm.
Um, uh, so, as a general rule of thumb, I let people know. I let people know that if you're dealing with a vaccine um, that's that's dealing with a bacterial pathogen, chances are very, very good that there is no ethical compromise in its history. Now, I still look into those, and occasionally you do find one that involves the use of fetal cell lines, but not nearly as many, not nearly at the rate that you do find in the development of viral pathogens for vaccine use.
And then I saw an article on your website at one point where you were replying to the whole list of products. I believe it went kind of viral on the internet a few years ago, saying things like
Mm-hmm.
Advil, Tums, Sour Patch Kids, all sorts of different foods and medications were connected to fetal cell lines. And you had a great reply to
Mm-hmm.
that. Can you give that to our listeners? Because I know that that was something that I think I avoided candy corn for about 2 years because it was on some list somewhere of things that were connected. And I'm very glad to be able to have it again. So can you just kind of give that answer to our listeners to kind of clear up those misconceptions that are out there?
There's a lot there, and I'll start by letting you know what started this whole thing, and this was during COVID.
Mhm.
Because we were one of the organizations that got underneath the scientific literature and said, hey, wait a minute, all of these COVID vaccinations, all of the ones that are being evaluated by the FDA for use in the United States, all of them are ethically compromised. Now, there was some argument as to whether or not that was true. And interestingly, some of those arguments persist to this day. Like if you were to call Novavax and ask them if aborted fetal cells or aborted fetal tissue was used in any way, shape, or form in the development of their vaccine, with a straight face, they will say no. And I can point to a paper where half of the authors listed on the masthead were employees of Novavax, and Novavax, in part, funded the the experiment in question, where the use of HEK 293 to evaluate a variety of different, they used it to express a variety of different versions of the virus, so they could test their vaccine across a spectrum of different spike proteins. And it said so right there in black and white in a peer-reviewed paper. I believe it was published in Cell, which is a highly thought of peer-reviewed publication. So it's really not arguable. They wrote it down. So you have situations here where the Greek dramatist Aeschylus in the 5th century BC was the first one to say that in war, truth is the first casualty.
Mm.
And truth was definitely a casualty during COVID because everybody had a point to make, everybody had an axe to grind, everybody wanted to make sure that they felt better about themselves and somebody else felt about them. The whole thing from a high level, from a high view of human behavior, was really kind of disgraceful. And of all people, a Catholic priest wrote an article on his blog. And blogs being what they are, they're not immediately notorious, but at some point people started to notice. He wrote a piece on his blog, and it was titled to the effect of if you're having a problem with the vaccines, or if HEK 293 is no longer ethical to use in modern medicine, then you need to kiss modern medicine goodbye. His point was that it's everywhere and it's everything, and we need to get over it and we need to move on. But and then he started throwing some examples out, and he partnered with a medical doctor that helped him with the research. And he put a list of medications at the end of that blog post that got a lot of attention. And he put things on there like aspirin and Tums and Preparation H. He put a couple of vaccines on there that actually were developed using aborted fetal cell lines, but a broken clock is right twice a day. And Tylenol and ibuprofen, a lot of things that are very commonly used. Claritin was another one. And that was brought to my attention. A number of people that are regular readers of our website came across that, and they were very, very concerned. So emails started coming in going, can this be right? And it's like, no, I don't think so. But let me look into it and I'll get back to you. And I started going through the list item by item. And he had originally put together a list of 14 medications. And out of the 14, I think there were 2 vaccines on there that actually had an association to abortion. The rest of them were a swing and a miss. Um, I'll give you a really good example. Aspirin. He said aspirin was developed with HEK 293, just like the COVID vaccines were. Aspirin was discovered. It was first produced in 1877 by a French chemist named Friedrich Gerhardt. 1877 was exactly 100 years before HEK 293 was available to the research community. So, aspirin being developed in aborted fetal cell lines or tested in aborted fetal cell lines or even sharing the same time and space with aborted fetal cell lines is an impossibility. So that was one. Acetaminophen, also known as paracetamol, was only 10 years younger than aspirin. It was first synthesized in 1887. So another one, it's impossible, absolutely impossible that you can make the equivalents. COVID vaccines, aspirin, or Tylenol can't happen that way. The most preposterous of the bunch, though, was Tums. Tums is nothing more than calcium carbonate and sugar, and it was invented by a fellow by the last name of Howe, and he invented it in the basement of his Chicago home in 1930. And he invented it to relieve his wife's indigestion. And it's all calcium carbonate and sugar. And it still is almost all calcium carbonate and sugar, but they put other things in there like colorants and flavorings and those sorts of things. Um, but it's still no different than a piece of chalk, chemically speaking, or an eggshell, or anything made out of limestone, because limestone is calcium carbonate, or the White Cliffs of Dover. So I started thinking about that a little bit, and I go, that has to be one of the most. You got to have a lot of sand to put that one out there, or you don't know a darn thing about chemistry. It's just like, well, they were developed with aborted fetal cell lines, so therefore anything made with calcium carbonate is ethically compromised. The state capitol building in Indianapolis, Indiana, is made out of limestone. I don't have a problem with that building.
Is it
Um,
possible that some of these medicines, not maybe not Tums, but things like Advil or other ones, could have been tested on the cell lines after they were developed for certain reasons? Because it seems like there could be a confusion if you take something that was developed on its like ethically, and you could do almost anything bad with it later on, and it's not going to change the ethical like nature of that item. You're going to just do something bad with it.
that's exactly correct, and that's an excellent point, because a lot of people started making that argument. And when I part of my research, when I was going through that list, I went to the sources cited by the medical doctor that was helping this priest put that list out, um, and found some interesting things, like the reference that supposedly proved the Tums were ethically compromised mentioned calcium carbonate, but it had nothing to do with Tums. It wasn't funded by GlaxoSmithKline, who now owns the brand for Tums. It was using microstructured or nanostructured calcium carbonate as a gene insertion tool, um, uh, for genetic applications, for genetic research applications. So it had nothing to do with Tums. It just had a chemical similarity to something. So if the argument were to hold water, then in fact the state capitol building in Indianapolis, Indiana, would be ethically compromised. Um, but it's a fair point. You have something that's developed ethically, and at some point in the future, some researcher, in a completely unrelated way to the manufacturer of that particular preparation, does something that involves aborted fetal tissue. Um, the question becomes, does that subsequent act then taint the origin of the drug? And the way I look at things, that's not, I'm a mathematician, that's not commutative. Um, you know, Henry Ford there cannot be held responsible for drunk driving, nor John Moses Browning be held responsible for armed robbery. It doesn't make any sense.
Not that this would scientifically happen, but if you took chocolate and then you tested it on a cell line for some reason, that doesn't make chocolate bad. It just means that that action over there that someone did is bad.
Exactly. The action, the subsequent action has no effect on the origins of a drug. Um, but that was argued. That was argued for quite some time, and I still think some people want to go back to
And
that.
I think that's a really important thing for people to be aware of, that there's a difference between how something was actually developed, like what went into the actual putting it together, and then what people do with it subsequently. Because that's a really important distinction to be able to make, is recognize that people can do bad things with a good object. But what you need to look up in
Absolutely,
this is, how is that object made?
that very well said. I think that is a very very important distinction, um, and it also brings into the conversation the difference between research and clinical testing and preclinical testing. If you're a drug manufacturer and you get the FDA's thumbs up that says, okay, you can sell this now, the last thing you're going to do is to do anything that's going to put you back into that approval cycle, because it's long, it's expensive, and you're never really sure you're going to recover your investments from a business perspective. So it makes no sense for a manufacturer that's been manufacturing ibuprofen. And ibuprofen was also on that list, and ibuprofen was ethically developed. There's no question about it. It would make no sense for a manufacturer who's selling ibuprofen, either on a prescription basis or over the counter or both, to quote unquote reformulate that medicine and use aborted fetal cell lines and throw it back into the approval process. It makes no sense from a business perspective whatsoever. So, um, understanding the difference between your research, which is what the calcium carbonate argument was for Tums. It was a gene insertion technique using nanostructured calcium carbonate. Had nothing to do with Tums. Had nothing to do with anything other than what the paper was about. That's pure research. Clinical testing and preclinical testing are what we're concerned about, and the development process. So in my mind, it is perfectly reasonable, if not obvious, that those two bodies need to remain separate. As you put it very well, people can do bad and evil things with something that was properly and ethically
When
produced.
you talk about research in that context, you're talking about something that already exists and using it for further research, right? Not the research that goes into developing it.
Exactly. It becomes an ingredient. It becomes an ingredient as part of an experimental design for something else.
But obviously, if you're developing a
Um,
new product and the research at the top is flawed, then that's going to affect the subsequent production of a new drug, correct? So, like, if the research is flawed when you're developing a new drug, that would be like the drug wouldn't exist except for unethical research. So, that would be the research in that sense would be a different side of things. Am I just missing what you're saying?
that's correct, and that's a test. My wife and I discussed this a lot, and there were people who wanted to make a distinction between a dependency on aborted fetal tissue versus an immaterial use of it. Um, and I'm not sure what that I'm not sure what that immaterial use of fetal tissue or fetal cell lines would be. But let's just say let's just accept the distinction for a moment. If a drug were to be developed, researched, tested, and brought to market, if that process right there depended on the use of fetal cell lines or fetal tissue in any way, shape, or form, And I think that's what creates that's what creates an undeniable ethical fault. The ethical compromise is associated with the dependency, with the critical dependency on the use of aborted fetal tissue or fetal cell lines. And that seems to satisfy just about everybody. And that but for test, if you look at, say, for example, the COVID vaccines, but for the use of aborted fetal cell lines, they would never, ever have been able to synthesize the spike protein. So that settles that argument.
Okay. And then where does the food industry fit into this? Because I know that there's, I think, artificial flavoring in certain products that doesn't have obviously the fetal cells in it, but was the science going into that artificial flavoring was developed using fetal cell lines. Can you talk a little bit to that as well?
Yeah, and that's um, if I were to look at the number of emails that I get over the course of a month and separate them into piles, that might be the biggest pile. And it's just kind of understandable. I mean, vaccines are a big deal because they affect just about everybody. And from a parent's perspective, they affect our kids. So we tend to have a great deal of interest and a visceral response to yet another vaccine. Oh, by the way, um, were you aware that if from birth to the age of 18, if you follow the CDC recommendations for vaccines, you would get stuck 98 times?
Wow.
Yeah. I'm glad I was born in 1959 because if I were born today, I wouldn't make it because I don't like needles. Um, but it's really remarkable how, let's just use the word reliant we have on, we've become as a society on these sorts of things. And it continues to grow because now we need to add the RSV vaccine and a couple of other things. But anyway, sorry for the hijack. But let's get back to the question. The use of aborted fetal cells in flavor enhancers and that sort of thing might very well be the biggest pile, because people come across that and say, "My goodness, I just ate a bag of M&Ms. What have I done?" Um, and there are websites and organizations out there that are, in my view, irresponsibly telling everybody that it's in everything.
Okay, for
Um,
clarification, do M&Ms have this? Are they tainted?
no,
Okay.
they do not.
I figured that was an example, but I just wanted to clarify for myself and for our listeners.
I literally have gotten those questions. Are my M&Ms okay? How about my Skittles? I love Jelly Bellies, and it's just like, yeah, I get all that. I mean, it would be horrifying to think that the foods we enjoy are connected to abortion. So I understand why people get so exercised over it. But the this goes back to about 2010, 2009, 2010. There was a company in California. I believe they were based in San Diego. A company's name was Senomyx. Um, and they came up with a method that could fool the tongue into thinking it was tasting something that wasn't really there. Um, and the flavor enhancers that they developed, they tested them in aborted fetal cell lines. And what they were looking for was the cell's response, producing specific proteins that our taste receptors on our tongues would produce, for salty flavors, bitter, sweet, in anything like that. So you could create the illusion that a food has high salt content when in fact it doesn't. And they were looking for the cells to express certain proteins, and then they would use human testing to confirm it, and then they would bring it to market. But the front end of that testing cycle had to do with aborted fetal cell lines. And it's just the testing. It's not part of the production of the flavor enhancers, which is really important. There are people out there that say, well, it's in our food. It's not.
But that's where it goes back to, because the research that led to these flavor enhancers was unethical. It makes kind of the whole line down after that unethical when it comes to
Yes,
efficacy. But then the actual products in the food themselves are not the fetal cell lines.
that's correct. But those flavor enhancers failed the but-for test. But for the use of fetal cell lines, they would not have brought it to market.
Okay.
I mean, it was essential. It was on the critical path to them developing what they developed. Um, and PepsiCo was one of the first companies to say this is kind of interesting. Now, I don't think PepsiCo walked in and said, "Hey, you're using aborted fetal cell lines. We want to try your stuff." I think um, objectively speaking, let's forget the ethical compromise for a moment. But objectively speaking, that technology is really really attractive. The artificial sweetener thing goes right out the window. We don't have to worry about aspartame or any of the other artificial sweeteners being carcinogens. Uh, the illusion of sweetness can be created by these enhancers in very, very, very small amounts. Salt intake could be cut down. All sorts of benefits could come from something like this. So Pepsi was initially interested. Debbie Vintage, our founder, um, learned of this. Somebody had alerted her to Senomyx and one of the patents that they filed, and also the language on their website. They were very open about the fact that they used fetal cell lines. Didn't hide it from anybody. It was there for anybody to read on their website in unambiguous terms. They were completely unconcerned. They didn't think that would create a problem from a marketing standpoint. They were wrong. Um, when when Debbie got people involved and started circling the wagons and started talking about a boycott, people started acting on that sort of thing. PepsiCo um, uh, started paying attention very quickly because it affected their market share very, very quickly. It's like, how dare you? And you can imagine a lot of the things that were said. How dare you put dead babies in my soda pop? That wasn't the way it was, but I think we have to understand kind of the visceral reaction, our gut level reaction to something like this. Like, I'm not sure I would have said anything different. Um, so PepsiCo, even though they were under contract, they continued to pay Senomyx, but they said we're not going to use any of your stuff. They went out and hired real people to do their taste testing. Campbell's Soup was also part of the front end that expressed interest in this technology. And when they learned of it, when it was brought to their attention, they were very quick. Nobody had to talk about boycotting Campbell's or anything like that. They were very apologetic. I think, in my opinion, Campbell's did a terrific job of kind of crisis management on the front end. Own up to it, say we're sorry, we're never going to do this again. That's not in keeping with how we view our products and our customers, and we're terribly sorry. It'll never happen again. Um, Kraft Foods was much more quiet about it, but they also decided not to proceed because of the visibility that Debbie Vintage and Children of God for Life created in 2011, 2012. That's chronicled in a very detailed way on our website. If anybody is curious um, about the history associated with that, it's actually a pretty good read.
Yeah. No, I read a little bit about that one. But like I said, when I was doing research for the ebook we wrote. And actually, that's a good jump. Okay, let me finish up this topic first before I ask another question. So are there any food products whatsoever that people would buy at stores that are connected with this at all? Did any companies go through with it?
Um, there are some, and some they sell their products here, but they're manufactured elsewhere. Ajinomoto uses these flavor enhancers in a number of their products. Um, and I think in part because it allows them to enhance the saltiness of a lot of their products without high sodium content.
And you have a list of that on your website, I believe, right? Some of the foods to avoid.
Yes,
Okay. I can always link that in the
yeah,
description for our viewers as well, so they can go and look at individual products.
right. And Cadbury Adams, Nestle's refrigerated coffee creamers. are also on that list.
Interesting.
But if you approach Nestle's with it, they say we've never done anything like that. It's like, I'm sorry, but that doesn't match up what's been written down in the past. Um, and Nestle has kind of a checkered history associated with the exploitation of the killed unborn. Um, and time permitting, we can probably touch on that in the development of infant formula for neonates, neonatal infant formula.
Hmm.
What they did to develop that was horrifying. Um, with aborted children, they were these children were delivered live, and what they were measuring was the transfer of amino acids through the umbilical cord to develop ultimately neonatal infant formula. And the nature of the testing resulted in the death of every single one of the children that were involved in that. And this is infant formula that was developed on the backs of murdered and exploited children.
Wow.
Yeah. And Nestle funded a lot of that. So I can understand them being a little hand shy because I mean it's not widely known that they did this, but the people that know won't let them forget.
I think most of these
Um,
topics are kind of generally known, but not widely known among most groups. And when they
I
are
agree with
known,
that.
there's misinformation going on, like with the article from the priest that was listing things that weren't connected with it at all. There's so much misinformation floating around.
Yeah, and at some point you have to wonder about what motivates that sort of thing because while some of this information is not easily discoverable,
Hmm.
um, you can look at a source like Wikipedia and look at a date and say 1877 is way before 1977 and be done with it. Um, but you're right. Um, I think the common person today has a very limited, if any, idea that aborted fetal tissue is used in producing medications or is used in this kind of human developmental research.
Hmm.
There's a project right now called the Human Cell Atlas, and they're trying to understand, among other things, when human cells start to differentiate. That's one of the things that makes stem cells, on one hand, so interesting and potentially dangerous because stem cells, they start out life not being much of anything other than a human cell. And something happens and this stem cell over here says, I'm going to be a brain cell or I'm going to be a neuron. And they develop along those paths, and that happens in generally the same way for every single human being that's ever been born or ever will be born. So this project gets at trying to understand human development from that point of differentiation and the growth of the organism to birth. Um, and if you think about what happens in 9 relatively short months, I know if an expectant mother isn't going to agree with that, but what happens in 9 months is nothing short of miraculous. Where you go from where you go from zygote to a fully formed human being with all of the parts that are working in concert and everything. And it's really it's an extraordinary thing, and the rate of development is rapid. Um, and they're trying to understand that. Among the benefits that they say associated with this fetal cell atlas is that they'll be able to identify and cure virtually all pediatric diseases, um, which sounds more than a little too ambitious. But yeah, it's but how are they going to do that? They need fetal tissue at every point along that developmental line. Um, and some of it undoubtedly the source will be aborted fetal tissue.
Can I go back
Um,
for a moment to what you were saying about Nestle's formula? Is any of that on the market today?
the neonatal
Yeah.
formula, yes, it is.
Okay. I just was curious. It was something that occurred to me as you were talking that I didn't ask about that. So that's good to be aware of. So anyway, sorry.
Yes. Yeah, and when people start to learn about that, I think they get angry, and I think that anger is justifiable.
Yeah. Like, because you're
Um,
killing children to help other children. And that is just so, like you said, is the irony. It's not like killing children to help other people is ever good either, but there's just something really that jumps out to you with that of
that is the murder and the exploitation of the unwanted children for the benefit of the other ones, the ones that are wanted.
can we go
And
into oh sorry.
no, no, please go ahead.
The last like so we talked about vaccines, we talked about the food element of it. Can you talk a little bit about cosmetics? Like I said, we did an ebook on the whole subject, which I'll link for our viewers if they're interested in learning more about it. But can you talk a little bit about the use of aborted fetal tissue in cosmetics and what's going on there?
Um, there's it's very difficult to know for sure
Okay. Why?
because that's relatively unregulated. Um, so to the extent that we know anything is because the manufacturers themselves tell us. And um, there's a company in particular called neo that is kind of the Senomyx of the cosmetics world,
Mhm.
where they make no bones about it. They openly disclose it. On one hand, it's kind of sad that people think that's no big deal. On the other hand, I I appreciate their honesty,
Mhm.
um, because are there other people that are using fetal tissue in ways that we don't know about? Yes, absolutely. Because they're not compelled to tell us, and they're not going to tell us because they expect it to be bad for business.
Is it unregulated? Like, so I'm guessing that what happens with drugs and medication that you said you can find it, and with food is that's all regulated because of like the Food and Drug Administration or the science along those lines. But then cosmetics aren't regulated. Is that why they aren't obliged to tell us?
Um, that's essentially correct. It's to the degree that they're regulated. There is the FDA does keep tabs on, generally speaking, cosmetics and personal healthcare products, but not to the degree that they look at medications. Um, those are two entirely different things. So what they're interested in is content of prohibited materials, safety. Is the labeling correct? Those sorts of things.
Okay.
In terms of how you made this hand cream, exactly. Please, we're busy enough. Don't bother us with that. So, it's not quite the wild, wild west, but it's much more the wild, wild west than medicine certainly are. And Neocutis uses proteins that that were ultimately sourced from an aborted fetus in a number of their what do they call it? Age-defying creams and that sort of thing, wrinkle creams and those. And for for skin repair and to improve somebody's youth, or to create a youthful appearance, because human cells taught them something, I guess. That's what makes these aborted and these aborted and exploited children valuable to medical science and commerce. It's their very humanity. Now, for all of the people that say, "What are you worried about? It's just a clump of cells," no, you can't own both sides of the argument. The only reason they're used is because they're human. So that argument is over. Um, and if you ask a microbiologist that question, why are fetal cell lines so important? That is the answer that he will give you. It may not be quite that clear, but it all comes down to they're human. Their very humanity is why they're important to their work.
Such a big issue that there's so much like to unpack in it. I might actually split this episode into two because there's so much here, but it's so
Oh yeah,
yeah, there's so much to unpack.
it, if it's just like, well, if we exploit an aborted child to give you this medicine that'll make you feel better or live longer, I would say I don't need that. I don't want to live longer if that's what it takes. So the can you do it should not be the question. It's should you do it.
Are there ethical alternatives to this in general in the medical field, or are we kind of reaching a point where this is so ingrained in this quote unquote science that it's going to be hard to, like, is there a way to develop these types of, like, the viral vaccines or other types of medicines that don't involve using aborted fetal cells?
Absolutely. Um, all of the ones, all of the COVID vaccines that are approved for use are ethically tainted.
Mhm.
But if you look at vaccines that are available elsewhere in the world, there are a great many of them that did not use aborted fetal cell lines. There are other cell lines that have generally the same expression characteristics. Now, remember, that's what cells do. Um, some of the proteins that are produced within the cell are called antibodies. They're proteins. And non-human primate cells, like the Vero cell line is African green monkey. Uh, the expression characteristics of those cells have been modified to a point where they very, very closely mimic the behavior of human cells. Um, there are insect cell lines. Caterpillar. They're caterpillar cell lines that have very, very similar expression characteristics. They are often used. There are adult cell lines or adult cell strains that have the same, almost the same level of utility, but they're argued against because they say, oh, oftentimes they're infected by, they have these legacy infection fingerprints. It's like a person's been sick. There's something in the cell chemistry that reflects that sickness, and that could mask and that could mask its reactivity to what we're doing. But fact of the matter is there are many, many alternatives to aborted fetal cell lines in developing virtually everything. Now, the exception to that is things like the Human Cell Atlas, where if you're going to study how organs differentiate, you're going to need those cells. And they're going to have to come from a human being. So that's one of those. Do we really want to answer that question or not?
Not definitely.
I
Yeah,
don't know.
not unless there's
It's
another
like
way
to
to
me,
do it.
yeah, to me it's clear. It's just like I'm okay never knowing that. If what it takes to know that is that horror,
Yeah, that's so true.
I'm okay with never knowing that.
So what should our listeners do from here going forward? Do you have any suggestions for them about how, like, I know that they can petition vaccine companies to try to make ethical versions of vaccines. What suggestions do you have for listeners after learning all of this?
I've been giving this some thought for a while because it's, this is one of these knowledge without application really isn't worth a whole lot kind of questions. And, I think the pro-life community in general, I think we can have the most significant impact in the shortest amount of time by ignoring what's in our medicine cabinets for the moment and thinking about what can increase awareness in a broad way. And the piece of legislation that we were working with Senator Bob Hall on in Texas last year, I think is a really good way to do that. Now, we worked with our state legislators for a reason, and I'm sure your listeners and I'm sure you are as well. You've seen California Proposition 65 warnings on things in your house, yeah, because it's on everything. It's on everything.
In California, this causes cancer.
Yeah, yeah. And they're on pillowcases, and it's like really, but that was a state law that affected everybody.
Yeah.
And if we take the same approach, um, and we get this passed, we get a law that requires the labeling of products that anywhere along the value chain used aborted fetal tissue or fetal cell lines or fetal material in any way, shape, or form, anywhere along the value chain, from just kind of proof of concept to stuff on the loading dock. if that has to be disclosed at the point of sale, then I think we can change things.
Yeah, because then
Just like,
people won't buy it, and then it's automatically boycotted.
right. And there have been other approaches that have been suggested, like this is called the Human Cell Product Labeling Act, and that's been introduced and actually hasn't made it to the floor in most cases, but sometimes it's been voted down. And there's model language for that legislation, and it would be better than nothing, but maybe not much better than nothing, because it's human cells. There are lots of cell lines that originated from adult donors that gave consent, and there's nothing wrong with that. Um, so I really don't need to know if whatever medicine I'm about to take was there was an adult human cell line involved. I don't care. There's no ethical problem with that. And it wouldn't be point of sale. I think if you're looking at a product in the store and you're looking at the label, this is like, you know what? Now I'm going to put this one back, and this one right next to it is different. I'm going to buy that one.
Yeah.
You change consumer behavior based on that knowledge, and Pepsi succumbed to the threat of a boycott in a relatively short period of time with the Senomyx situation. That happened very, very quickly, and it was all grassroots. And it was, it was a mom from Florida that said, "I'm going to make your life miserable until you change." It worked.
Yeah.
So if we focus on dealing with our state level representatives, saying, "Look, we really need something that discloses this sort of thing at the point of sale," because we have cosmetics out there that say no animals were harmed by this product.
Mm-hmm.
Okay, why can't we have no human beings were harmed? No unborn children were harmed. Um, that seems comparatively like a money shot. It should be relatively simple to do.
Yeah.
And if we focus on something like that, that generates a great deal of awareness in a relatively short period of time, people standing in the pharmacy or standing in the grocery store or standing wherever, they will have that information available before they make a purchase decision. We can make a difference very, very quickly.
And not only will the companies that are using these products lose sales, the products that are doing things well will have a boost in sales. So it'll be like a plus to them for doing things right. And then you'll be able to hopefully stop the use of it in products for the other companies.
Exactly. It'll change behavior. And those alternatives that we talked about just a few minutes ago, they'll start using them.
Well, this has been such an interesting conversation. It's something that I've been like coming to learn more about over the past 5 years or so. And it's really good to have a source to go to that. I know that you guys are doing the research well and having a very logical approach to it, not just saying, oh, this good product was tested on it by some person and some bad thing, so the whole product's tainted. You have a very practical and ethical approach, from what I can tell, to looking at this issue. So thank you for that.
Oh, it's as we like to say, it's the practice, not the product. Um, so it's the human behavior around the product, and a lot of times we end up focusing on the wrong things, and that's just a part of human nature, I guess, because we want to be able to vent our anger at something, um, like that evil corporation. I mean, I've worked for corporations. I'm here to tell you, corporations have no feelings. Um, they just don't. Um, a corporation's job is to grow and make money.
Yeah.
Um, and hopefully in ethics, business ethics has an awful lot to do with that. But we need to focus on the behaviors as opposed to the things.
And stopping the behavior of using this research in for developing products is the first way to start. So, yeah.
Yes, and with sufficient awareness, I think that could be largely organic.
Yeah. Like you said, we have the products that say no animals were harmed. We need to be able to, as pro-lifers, ask for those kind of products to be labeled as well.
yes, no aborted children were exploited, or something along those lines. I really think that we should put our efforts into something like that, because the focus, and again, it's perfectly understandable. I'm not trying to be critical of anybody and how they approach this sort of thing. Um, because I've asked those questions of myself. I just go get the answers myself. But we tend to be focused on what's in our medicine cabinet.
Mhm.
What do I throw away and what do I keep? Okay, when you're done with that, your medicine cabinet is half empty, but the problem is still there.
Mhm.
And the problem, there are alternatives. There are alternatives. There are people working on alternatives every single day. Um, Interestingly, we talked to my wife and I had a conference call with a pharmacologist, PhD in pharmacology, and we asked her that question. It's just like, well, it seems unnecessary. Why is it so prevalent? And she said, your average researcher is lazy. They do it because it's an accepted standard. If you do it that way, no one's going to question it. So it's the easy button. It's the easy way out. Um, that's the way we've always done it. You know, all those things that are just kind of excuses.
Well, thank you so much for this conversation today. I'm really, really glad that we got to have an episode or possibly two episodes on this topic.
Well, there's a lot of meat on that bone, but unfortunately, uh, but let's get our heads around it the right way, and let's make a difference.
Yes. Thank you. And to all of our listeners, please like, follow, subscribe. I'll link the ebook too if you're interested in that for learning more about this topic on the cosmetics angle. And keep on living the culture of life. God bless.
God bless. Thank you very much.
Thank you.